The study of ancient hominins has long fascinated researchers and historians, and among these enigmatic figures, the Denisovans stand out. While they may not be as well known as Neanderthals, recent discoveries about Denisovans are shedding light on an essential chapter of human history. As scientists delve deeper into the mysteries of Denisovan DNA, they are unearthing insights that not only reveal the unique adaptations of this ancient group, but also highlight their surprising contributions to modern human genetics. This exploration aims to unravel the latest findings, emphasizing how Denisovans help us understand our own evolutionary journey. At the heart of this research lies a remarkable revelation: Denisovans have left a genetic legacy in contemporary human populations. Evidence suggests that interbreeding between Denisovans and early modern humans occurred, particularly among groups in Southeast Asia and Oceania. Genetic analyses indicate that some individuals today carry between 1% and 6% of Denisovan DNA in their genomes. This genetic inheritance is not merely a curiosity. It has real implications for traits like adaptation to high altitudes, as seen in modern Tibetans who possess a specific gene variant linked to their ability to thrive in such environments. These interbreeding events prompt intriguing questions about the nature of interactions between Denisovans and early humans. Did these encounters represent fleeting moments, or were they part of a more complex web of relationships that allowed for the exchange of genetic material over generations? The evidence leans toward the latter, suggesting that Denisovans were not isolated but rather engaged in a dynamic interplay with other hominin groups. To fully appreciate the impact of Denisovans on modern human genetics, it is essential to delve deeper into the mechanisms of interbreeding and the specific contributions these ancient relatives made. As researchers have sequenced Denisovan genomes from fossil remains, they have identified unique genetic variants that can be traced to Denisovan ancestry. This genetic legacy has important implications for understanding how certain traits have evolved in human populations. One of the most striking examples of Denisovan genetic influence is seen in modern populations living at high altitudes. Tibetans, for instance, have developed remarkable adaptations that allow them to thrive in environments where oxygen levels are significantly lower. Studies have shown that a variant of the EPAS1 gene, which is associated with efficient oxygen utilization, is prevalent among Tibetans and is believed to have been inherited from Denisovan ancestors. This finding illustrates how ancient interbreeding can have lasting effects on the physiology and health of modern human populations.
Furthermore, Denisovan DNA has been linked to other traits beyond high-altitude adaptation. Genetic analyses suggest that Denisovans contributed to the immune responses of certain modern populations, potentially enhancing their ability to combat diseases. This genetic legacy opens new avenues for research into the complex interplay between ancient genetics and contemporary health issues. Population dynamics and migration patterns. Emerging evidence indicates that Denisovans were not a singular population, but rather comprised multiple groups dispersed across various regions of Asia. This revelation has significant implications for our understanding of population dynamics during the Pleistocene epoch. The genetic analysis of remains from Denisova Cave, along with other archaeological sites, suggests that these hominins occupied a range of ecological niches and adapted to diverse environments. The mobility of Denisovans is further underscored by the discovery of Denisovan fossils in different geographic locations, including the Tibetan Plateau and the island of Luzon in the Philippines. These findings indicate that Denisovans were not confined to a specific area, but were instead part of a broader network of hominin populations. The implications of this mobility suggest that Denisovans engaged in extensive migration, potentially following herds or responding to climatic changes that affected their habitats. Moreover, the interactions between Denisovans and other hominins, such as Neanderthals and early modern humans, highlight a complex web of relationships that shaped human evolution. The evidence of interbreeding with Neanderthals has been well documented, and the genetic exchange with Denisovans adds another layer to this intricate narrative of human ancestry. Understanding these interactions is crucial for reconstructing the evolutionary pathways that led to the emergence of modern humans. Technological advances in ancient DNA analysis. The field of ancient DNA research has undergone a revolution in recent years, significantly enhancing our ability to extract and analyze genetic material from ancient remains. Advanced sequencing technologies, such as next-generation sequencing (NGS), have allowed researchers to recover high-quality Denisovan DNA from fossil remains and sediments in Denisova Cave. This progress has provided unprecedented insights into the genetic makeup of Denisovans and their adaptations to varying environmental pressures. One of the most exciting aspects of these technological advancements is their capacity to illuminate the adaptive traits of Denisovans. For instance, studies have identified specific genetic variants associated with traits that enabled Denisovans to thrive in harsh environments, such as cold climates and high altitudes. The discovery of the EPAS1 gene variant among modern Tibetans, inherited from Denisovan ancestors, underscores the adaptive significance of interbreeding events.